Now, the invitation by IUPS to give the Fenn lecture is the chief way in which the Union can honour an individual physiologist, and in this way today we honour Bernard Katz. He is firm that he is a physiologist, although his work, nearly all his life's work, was done in first a unit, and then it became a department at University College with the title Biophysics. Indeed, I at one time held a title of reader in biophysics, but I am equally clear that I am a physiologist. But this was the unit that was started by A.V. Hill, this same man under whom Fenn worked. And when he retired in 1952, Bernard Katz succeeded him as head of what then became a department of University College, and he remained head until he retired in 1978. Katz was born and educated in Leipzig, born 1911. His 80th birthday was celebrated 2 years ago. His parents had emigrated from Russia to Germany much earlier, but he was forced to emigrate once again by the Nazi anti-Semitism, and in 1935 he came to A.V. Hill's laboratory, this research department within University College, where a whole series of famous names from overseas came to work and to receive inspiration from A.V. Hill. Katz's best-known work, of course, is that about which he is going to be speaking this evening, on the mechanism of transmission of an impulse from a motor nerve to a muscle, and particularly the Quantal release of acetylcholine, work for which he received a Nobel Prize in 1970. But he made many other important contributions connected with nerve and muscle. There is a paper of his published in 1939, which remains a classic, about the response of a muscle to stretch while it is active. He worked with Steve Kuffler, again, alas, someone whose death we have to mourn, of a few years ago, in Australia. Work on the very interesting properties of muscles of crustaceans. He measured the electrical capacity of the surface membrane of muscles, finding it was surprisingly large, which had an interesting interpretation later on. And together with Alan Hodgkin, whom I mentioned earlier, he demonstrated the very specific role that sodium ions have in the propagation of excitation, both in nerve fibers and in muscle fibers. Well, now I will call on Sir Bernard Katz for his Fenn lecture entitled Neural Transmitter Release from Quantal Secretion to Exocytosis and Beyond.
Mr. Chairman, ladies and gentlemen, well, first of all, may I thank you, Andrew, for your very kind introduction, and I'm most grateful to you and your council for inviting me to give the Wallace Fenn lecture on this great occasion. I have pleasant memories of my meetings with Wallace Fenn, whom I greatly admired as a person and as a scientist. I'd heard very much about his achievements long before we met. I knew about him through our mutual friend and teacher, A.V. Hill, in whose laboratory we both had been working at different periods, and from whom we both learned a great deal about physiology, about nerves and muscles, and about many other things. The subject I want to talk about, neuromuscular and synaptic transmission, is one that has fascinated me and provided me with great fun and entertainment for more than half a century. I was introduced to the field at a time just before Henry Dale and his colleagues discovered the role of acetylcholine as the chemical mediator, as a transmitter from motor nerve to muscle fiber. It's probably difficult today to envisage the enormous impact, indeed the shock, which this new concept brought to physiologists. The prevailing idea in the 1930s had been that there was direct electric propagation and with some slight delay at the neuromuscular junction and at central synapses, neuronal synapses generally. They thought, the idea that a complex sequence of chemical steps should intervene at the contact between motor nerve and muscle, that is, between two electrical waves of activity, which appear to be a continuous and unfailing process, that idea seemed strange, and there was prolonged argument for some 10 years before Dale's idea and his powerful experimental evidence were generally accepted. And even after the acetylcholine story at the myoneural junction had entered the textbooks, there was a long interval before the idea of chemical transmission was successfully applied and was supported by convincing evidence in the central nervous system. I remember discussions at our Physiological Society meetings at which doubts were expressed whether the neuromuscular junction should be classified as a synapse at all. And this was slightly upsetting to those of us who had devoted ourselves to this junction in the belief that it was an experimentally favorable object whose study could throw considerable light on synaptic mechanisms elsewhere. Now, to work out the details of this process of transmission, to unravel the subcellular mechanism of its various steps, of the neurosecretory process in the motor nerve terminals, and of the subsequent chemoelectric transduction in the postsynaptic membrane, all this was a considerable intellectual challenge which induced my colleagues, Paul Fatt and later José del Castillo, Stephen Teslev, Ricardo Milady and myself, to set to work, making use of the intracellular recording technique which had just been introduced and which enables one to record localized events at the synapse of single cells. In the spring of 1950, Paul Fatt and I had completed a study of the end plate potential in the normal twitch fibers of the frog Sartorius. And we turned to a different preparation to try and explore the myoneural junctions in the so-called tonic muscle fibers of the frog. Now, these are fibers which do not produce propagated spikes and which only contract slowly during repetitive nerve stimulation. Such fibers are very rare or non-existent in the sartorius muscle, so we used the toe muscle, which we knew had a proportion of slow fibers. We experienced great difficulty in even looking for them, and we knew that the electric responses were likely to be of much lower amplitudes than at the end plate of twitch fibers, and as we turned up our amplifier gain to have a better chance of finding them, we still failed in our attempts. But what we found instead was much more exciting and proved to be of much greater importance. By simply turning up the amplifier gain, we discovered the miniature end plate potentials and decided to devote ourselves to this new and quite unexpected phenomenon. Now, may I have the first slide? Well, that's all right. Now, this is, in fact, taken from the very first experiment in which Paul Fatt and I observed what is shown on the left half there at the top, that is at slow speed and high amplification. These little blips, all of much the same time course, these are the miniature end plate potentials which went on without any form of stimulation at all. If we put our electrode was taken at the end plate itself. If we put our electrode on the right-hand side, in the same muscle fiber 2 millimeters away, we only saw a very much reduced and attenuated residue of these spontaneous little blips. What is shown in the lower part, there we recorded at high speed and low magnification, and we stimulated the motor nerve at the beginning of the trace, which at the end plate gave us that characteristic double-step response. The first step is the end plate potential, which is due to the direct effect of the transmitter acetylcholine. And 2 millimeters away we don't see it, and we only get the propagated spike, which appears a millisecond later in order because it takes that much time to travel 2 millimeters. Slide off, please. Now, well, so we decided now to devote ourselves to this phenomenon, which was really quite new. Well, it was almost new. I'll come to that in a moment. Well, now I must point out that this happened early in 1950. It happened at a time when nothing was known about the ultrastructural details of the neuromuscular junction or of any other synapse, nor was anything known about their subcellular or molecular activities. And we had a long way to go with our tentative interpretation of this event. Today we know that the miniature end plate potential is due to a sudden discharge from the nerve terminal of an all or none packet of several thousand acetylcholine molecules, which react with the postsynaptic receptors before being destroyed by the esterase in the synaptic cleft. We also believe that the discharge is due to so-called exocytosis, that is, the fusion with the axonal surface membrane and the momentary disintegration of a synaptic vesicle. But it took us some 4 years before we arrived at this conclusion, and it may not be inappropriate if I tell you, by way of anecdote really, something about the rather roundabout way in which we stumbled from one interpretation to another and changed our views in the light of evolving evidence. But before I go into this, I would just like to mention a preamble which is quite instructive. When Paul Fett and I looked at the miniature potentials dancing in their irregular rhythm across the cathode ray screen, well, I not only experienced a mixture of aesthetic satisfaction and astonishment, but I immediately recalled that I had seen these things 2 years earlier, but I had dismissed them as an artifact, and until that moment of rediscovery had completely forgotten about them. In April 1948, I was working on the muscle spindle of the frog. I was interested in the local process by which a mechanical stimulus, stretching the muscle, is transduced into an electrical change and evokes impulses in the attached sensory nerve. I used what was then regarded as a rather refined technique by isolating the small bundle of intrafusal muscle fibers together with the sensory axon. Well, one day, as I was recording from the surface of the isolated bundle, I noticed a spontaneous discharge of small electric blips, which had nothing to do with the sensory nerve, and whose timing was quite independent of the sensory nerve impulses. They looked very much like end plate potentials, with the difference that they were very small and spontaneous, that is, of unknown origin. Well, can I have the second slide, which, thank you. Now, I don't know whether you can see these little blips. Well, this was my prehistoric recording of miniature end plate potentials with external recording from a small intrafusal bundle. This was in April 48. These things were about a tenth of a millivolt in amplitude with that recording method, and they are going rather fast, but they were contributed to by several end plates in parallel. Well, I can have the slide off, thank you. When I gave a small dose of curare, this phenomenon disappeared, which seemed to confirm their identification as end plate potentials. Now, this was a very unexpected observation which should have made me stop and think, but at that time, April 48, I put it aside and completely ignored it. You may ask, why did I not pursue the matter then? I suppose the reason is that although it was puzzling, it was an unwanted diversion. It had nothing to do with my immediate research plan. What I wanted to investigate was the origin of the sensory discharge, and the events which occurred at the motor end plate seemed irrelevant. At least they had no obvious relation to it. Secondly, for this particular experiment, I had cut the motor nerve fiber, which I didn't need, and I had cut it in the immediate vicinity of the motor end plate. And this allowed me to satisfy myself with a facile technical explanation, namely that the spontaneous end plate discharges arose from an artifact. I thought that they were caused by damage to the motor nerve endings, that they presented injury discharges of impulses in the terminal branches of the motor nerve fiber, and that this would then, of course, cause the secretion of acetylcholine and produce recordable little end plate potentials. And that permitted me totally to forget this phenomenon until I was suddenly reminded of it 2 years later. Now, when I saw my little spontaneous blips again, this time Paul Fett and I decided to carry out a thorough investigation of this accidentally rediscovered phenomenon and to make a proper job of it. We ascertained that they were a normal phenomenon and not caused by any injury of the motor nerve endings. Well, the discovery of these miniature end plate potentials provided me with tremendous fun. Simply watching them flitting about on the oscilloscope gave me, and I must say it still gives me, an almost childish pleasure, which is difficult to convey in a sober scientific report. But it also exercised the more intellectual parts of our cerebral cortex for a good many years. What was the nature of these localized subcellular all or none responses? What fired them? What was the driving force behind their irregular discharge, going on in the absence of any known stimulation? Well, to call them spontaneous is a convenient way of indicating that the cause of the phenomenon is unknown, but beyond that it gave us little satisfaction. Well, it took my friends and me many years to obtain answers to some of these questions. We know now that similar miniature potentials occur at a wide variety of synapses, possibly at all those junctional contacts between nerve cells where chemical transmitters operate. We also know that they represent the basic coin, the Quantal units by which chemical transmission works. The large normal end plate potential which one records in a muscle fiber after the arrival of a nerve impulse is made up of hundreds of miniature potentials. Today, the most widely accepted view is that the Quantal miniature blips are due to the discharge of the transmitter content of a synaptic vesicle by so-called exocytosis. But as I said, it took us several years to arrive at this interpretation, and it might be of some interest. There may be some lessons to be learned from our stumbling earlier attempts to explain the occurrence of these little spontaneous all or none potentials. Thank you. It clearly was a challenge. We were confronted with an unexpected new phenomenon, a subcellular all or none event, similar in some ways but quite different in others from the classical full-size impulse. Well, my first attempt to interpret the miniatures I've already mentioned was simply to regard them as artifacts, as a form of localized injury discharges in the terminal arborization of the motor axon, and so to dismiss and forget the whole thing for a couple of years. When we rediscovered them with the new intracellular recording technique, we had to think again. This time we considered the possibility that in extremely small cells or in extremely fine terminal nerve twigs, that Brownian motion, in other words, the random motion of ions across the membrane, might produce sufficient voltage noise to exceed the firing threshold very occasionally, and so to give rise to an impulse in a terminal branch. Well, this was quite an interesting idea at the time. You still had to explain why such an impulse should remain localized, and why should it fail to spread to other axon branches. But this was not very difficult to visualize, and we had in fact experimental observations to support it. We had come across other cases where an impulse in a small branch remained localized and did not flare up into an axon reflex. For example, I had observed spikes in the sensory terminals of the muscle spindle which remained localized. And we were quite familiar with the fact that when we dissected the giant axons of the squid at Plymouth, we quite often, on cutting a small branch, produced only a local twitch without firing the whole mantle muscle. Well, all this can be explained by the mismatch between a very small axon branch and its large parent stem. The small branch can't fire back into the main trunk because it doesn't supply enough current to overcome the impedance mismatch at the point of bifurcation. But then when we pursued our noise hypothesis quantitatively and tried to calculate how much noise voltage there could be in a motor nerve terminal, it seemed much too small by at least an order of magnitude, much too small to approach the firing threshold. And for this and for other reasons, we abandoned this interpretation. The next, that is my third hypothesis, centered around the concept of a carrier molecule in the terminal whose activation would be able to transfer a quantal amount of acetylcholine across the membrane. We thought this would occur spontaneously at a low rate and become vastly accelerated by the nerve impulse with the help of calcium ions, which were well known to be an essential cofactor. In those early days, Paul Fatt and I favored the idea of an ionic exchange between the terminal cytoplasm and the external fluid, in which acetylcholine rather than potassium was exchanged for the sodium ions which entered during the impulse, during the nerve impulse. Well, it was a somewhat fanciful modification for the special case of the presynaptic terminal of what was considered to go on in the main part of the axon. For a couple of years or so, we harbored the idea that the impulse might secrete acetylcholine in this way, but further experiments demolished the scheme completely. We found that the discharge of miniature potentials persisted even when there was no sodium in the outside medium and when all traces of electric excitability had been eliminated. Now, in 1954, we observed this in experiments in which we used an external medium that consisted of an isotonic solution of potassium sulfate, which sounds a bit rough. Well, this was many years before the famous pufferfish poison tetrodotoxin became available for experiments of this kind. Furthermore, by 1954, it had become very clear that the size and time course of the miniature potential was quite independent of all the presynaptic stimuli which we used. None affected the amplitude of the individual miniature potentials, although they might change their frequency of occurrence by several orders of magnitude. Now, this applied particularly to large changes of membrane potential which we imposed on the motor nerve terminal, either by direct polarizing currents or during the impulse. The size of the miniature potential was the same during rest and during activity. Now, can I have the next slide, please? Can you go one further? I don't know whether they're out of order. Yes, this one. This shows an example of, there are two double traces. In the lower trace, doesn't matter. In the lower trace, we show how we applied the current with the polarizing cathode near the junction to the motor nerve terminal, and you can see how the frequency of these miniatures, which was very low before and after, increased dramatically. But when we measured the amplitudes of these things, they did not change. We could increase the frequency of these of this background discharge of miniature potentials a few thousand times, without altering the magnitude at all. Can I have the slide off, please? Now, this was just one example of the various stimuli by which we raise the frequency without altering the amplitude. Anything we did to the presynaptic membrane would affect the probability of occurrence of these little blips, but not their individual size. Now, the constancy of the transmitter quantum under such widely varying conditions the fact that the quantal amount of acetylcholine ions emerging from the nerve terminal was independent of the electric driving force across the presynaptic membrane, we felt that this required a radical revision of our interpretation. It seemed to us now extremely unlikely that the transmitter quantum could be discharged in the ordinary way by ionic flux from the cytoplasm across the surface membrane. Because if that happened, the intensity of transmitter release, and therefore the size of the miniature potential, would vary with the presynaptic membrane potential. The constancy of the quantal packet suggested to us that the acetylcholine was prepackaged and was parceled up within subcellular particles from which it was discharged into the outside in an all-or-none manner. Now, shortly after Del Castillo and I reached this conclusion, we became aware of the discovery in the electron microscope of the presynaptic vesicles, discovery which was made in 1954 by Paley, Palladio, De Robertis and Bennett. We are very excited by these reports, particularly when we saw David Robertson's pictures obtained on motor end plates of the lizards. Now, from what we knew about the miniature potentials, about their rapid time course, and the short latency of their release after a stimulus, we proposed a hypothesis that was further elaborated later, but has remained substantially the same as we presented it some 39 years ago. We argued that the acetylcholine package, confined within its vesicular bag inside the terminal, had to pass through two membrane barriers before being released into the synaptic cleft and so becoming free to diffuse and react with the postsynaptic receptors. The package had to be freed both from the vesicle and from the axon. And to do all this in a small fraction of a millisecond, a simultaneous mutual reaction of the two confining membranes, that is, binding, fusion, and opening of the two barriers simultaneously, was required. The critical site of this reaction, we argued, must be a special molecule on the cytoplasmic surface of the cell membrane. Now, can I have the previous slide now? Thank you. Well, this is in fact yes, well, this shows a freeze fracture picture of nerve terminals facing the electroplaques. That is, these are, as you know, are modified neuromuscular junctions which are forming the electric organ. And you can see in this nice freeze fracture diagram, you can see the vesicles in that the nerve terminal, and here and there one sort of opening up in the way in which we are hoping to see at the neuromuscular junction, but at that time I hadn't seen it. Well, the evidence was greatly strengthened. Slide off, please. The evidence was greatly strengthened by the striking experimental observations of Heuser and Ries and their colleagues. They managed to fix end plates by a rapid freezing technique at the moment when the nerve impulse was producing a very powerful quantal release. They made use of agents that potentiate transmitter release, employing substances like aminopyridine or tetraethylammonium, which lengthen the duration of the terminal nerve spike and thus prolong and intensify calcium entry, thereby powerfully increasing the number of quantal packets delivered by a single stimulus. And I want to show a few pictures from their work. Well, these are electron micrographs cross-sectioning the nerve terminal with its vesicle inside. The upper part shows what happens at rest. The lower part shows what happens when they catch it at the moment of most of an intensified quantal release, and you see things that look like exocytosis, like breaking open of vesicles. The next one shows another example of this kind caught at the peak of activity. And the next slide, which is a beautiful picture which they obtained with freeze fracturing, what happens here is they happen to slice it so that you are looking from outside onto the membrane of the nerve terminal, and you imagine that the volume of the nerve terminal, which the vesicles queuing up, is behind the screen. And at the top you see what happens normally when there are just sort of two rows of particles which may be associated with the calcium channels visible. And during the height of activity, you look into these craters of vesicles that have opened and have presumably discharged their quantal packets. Slide off. By relating the amplitude and the quantal content of the end plate potential to the number of vesicular openings, Heuser and Ries concluded that the single vesicle was very probably the individual container of the quantal acetylcholine packet. The most direct evidence for vesicular exocytosis has been obtained on secretory and on neurosecretory cells, where degranulation of the vesicular core can actually be seen in the microscope, and where the volume of the vesicle and its quantal size are very much larger than at ordinary synapses. At neuronal and neuromuscular synapses, evidence of the same directness has not yet been achieved. There are some ingredients which are still missing to put it on the same level as has been reached in degranulating mast cells or in the adrenal medulla. And we are still lacking cytological and macromolecular biochemical markers, and the method of measuring stepwise increments of membrane capacity has not yet attained the sensitivity and the time resolution which is needed to make it applicable to quantal release at ordinary synapses. At the neuromuscular junction, the vesicles are of the so-called clear or translucent type. There are no vesicular cores which can be seen to be bodily discharged from the cell, nor have vesicle-specific soluble protein markers been identified up to now, which would enable one unambiguously to distinguish vesicular and cytoplasmic transmitter release. And it's not surprising, therefore, that in spite of the very strong and suggestive evidence, the vesicular hypothesis as an explanation of quantal transmitter release is still subject to a certain amount of argument. The evidence for exocytosis has recently been strengthened by electrical measurements of the membrane capacity, which shows a sudden increment when the vesicular membrane is added to the cell surface. Once again, the most direct observations were made not on transmitter release from nerve terminals, but on degranulating mast cells. However, capacity changes have also been recorded from presynaptic giant terminals in the squid and from neurosecretory endings in the pituitary. Now, an interesting feature was that in all these cases, the response to stimulation was a sudden stepwise increase of capacity of the surface membrane, whereas the recovery, which one would expect to occur during subsequent endocytosis, was very slow, taking many seconds and often too slow to be detected at all. Now, in some respects, this picture that has emerged differs from what I originally visualized. I thought that the vesicular discharge would not only be a very quick process, but that it would be brief and rapidly reversible. In other words, I had supposed that the vesicle would discharge its diffusible transmitter molecules through a small transient fusion pore and then quickly become separated from the surface membrane and start reaccumulating acetylcholine from the surrounding cytoplasm. This does not seem to be the case. Even at the neuromuscular junction, the transmitter release appears to be associated with full exocytosis, in which the vesicle explodes, turns inside out, and becomes part of the axon surface and is recycled only very slowly over a period of many seconds. Why this should be so remains an unsolved puzzle to me. I can see that full exocytosis, with a wide opening and incorporation of the vesicle into the cell surface, that is necessary for degranulation, when the solid core of the vesicle has to be bodily discharged from the cell. But in the case of small diffusible molecules like acetylcholine, the transient formation of a fusion pore would seem to be more economical and indeed quite sufficient for transmitter release. Well, I sought the help of a colleague quite a number of years ago in our mathematics department, and I asked him how large a pore would one need and how long it would have to be open to allow some 99% of the diffusible acetylcholine content of a 500 angstrom vesicle to be released. Well, the answer was that a 50 angstrom pore, that is 1/10 of the vesicular diameter, would be enough to allow all the free acetylcholine to be effectively discharged in much less than 100 microseconds. Now, Wolf Almos has shown that transient fusion pores do indeed occur even in mast cells, where they often appear as a preliminary event to full exocytosis and degranulation. Nevertheless, the electron microscope studies by Heuser and Ries and by Ceccarelli, as well as all the existing electrical measurements, indicate that the normal process of neurosecretion is not due to a brief reversible pore formation, and that it takes a long time to recover. To these should be added the exciting recent experiments by Bill Betts and his colleagues, in which they followed the fluorescent staining of what appear to be recycling vesicles. All this suggests that the reappearance of a vesicle after exocytosis is slow. However, I still think this is a problem that needs further study. One might possibly argue that the conditions under which neural exocytosis has been observed are extreme and involved an excessive sorry. What have I done? Oh, I see. Well, it doesn't matter. Thank you. In fact, I don't need it anymore. Well, what I said is that one might possibly argue that the conditions under which neural exocytosis has been observed involve an excessive form of stimulation, which may somehow overload the system and lead to departures from the normal kinetics of recovery. But if you do that, this would be a kind of special pleading against the existing evidence, which all points the other way. Now, if we accept the evidence for very slow recovery of exocytosis, some interesting consequences ought to be considered. The structure of the active zones at the neuromuscular junction indicates that there's a limited area of reactive membrane sites and a limited number of closely adjacent, that's presumably of immediately dischargeable vesicles in the motor nerve terminal. A large junction in a forward muscle has several hundred active zones with vesicles lined up like twin cartridge belts, about 50 vesicles at each zone. Now, can I have the next slide? This came from Professor Couteau's laboratory, in which they just sliced, in which they, well, I think you can see it without the pointer, actually. This is a very interesting gadget, yes. Well, you can see here a part of one of those double cartridge belts just facing the electron microscope. And then the next slide, which shows a schematic diagram of the way in which these vesicles are lined up immediately alongside the active zone in the postsynaptic membrane with its folds, and the neuromuscular junction is shown below. All right, now there are, as I said, a few hundreds of these active zones, and each containing about 50 vesicles lined up in a way that one might imagine are immediately available for release. Well, this gives a maximum of some, slide off please, a maximum of some 30,000 to 40,000 vesicles along these zones, and this would be the absolute maximum number of quanta which might be regarded as available for immediate release. Actually, only a small fraction of them would be immediately available, because exocytosis clearly would require not simply close membrane contact, but the apposition of the specific fusion-competent protein molecules at the two surfaces. Now, all right, once a vesicle has discharged its content, according to the existing evidence, it remains in position, exploded and flattened inside out, and occupying an appreciable area of the surface membrane. And it takes at least several seconds before it is removed and before the previously competent reactive site can be restored. So this patch will presumably be in a refractory state for many seconds, unable to offer a competent fusion molecule to the next approaching vesicle. Well, it's known that after a normal full-size end plate potential, there is first a short period of facilitation, which is followed by a phase of depression of transmitter release, some 5 to 10% down, which recovers only slowly with a time constant of about 10 seconds. In the literature, this period of depression has been ascribed to the time needed for vesicles to move up and replenish the readily releasable pool of quanta. But I wonder now whether a more important factor might not be the time required for the recovery of the active membrane sites. The situation becomes even more problematic if we consider the case of small central synapses, where the few release sites present at an individual terminal, bouton, could become totally refractory during a burst of activity, unless membrane recovery from exocytosis is speeded up very substantially. Well, clearly further experimental exploration is needed on this subject. One of the questions which I'm asked occasionally is, what is the point of releasing a transmitter in highly concentrated packets from discrete points of the cell surface, rather than more diffusely from the whole of the cytoplasm? Well, I think at the neuromuscular junction, at least, we can give a very plausible answer to this question. A very powerful destructive enzyme, the acetylcholinesterase, is anchored to the basal lamina in the middle of the synaptic cleft, that is in between pre- and postsynaptic membranes. Well, this had puzzled me for a long time. It seemed a paradoxical arrangement to place a formidable enzyme barrier right in the path of the transmitter, between its source and its target. Now we know that when we block the esterase, the duration of the miniature current becomes much longer, it's more than doubled, but the initial amplitude of the current increases only slightly, maybe up to 30%. We've also found that normally the acetylcholine molecules are hydrolyzed by the esterase so rapidly that they can react only once with a receptor and cannot repeat their action. In other words, the duration of the normal miniature current barely exceeds the lifetime of a single acetylcholine activated ion channel. So we have here the peculiar situation where the local esterase sitting in the synaptic cleft rapidly destroys the transmitter after it has produced its single brief action, but it does not prevent its initial access to the receptors. The reason why the system works so well, in spite of the interposition of the esterase, is entirely due to the Quantal process of secretion. The transmitter is secreted instantaneously in a highly concentrated packet containing several thousand molecules. At the moment of release, the local concentration of acetylcholine in the synaptic cleft is much higher than necessary to saturate the local enzyme barrier. Consequently, most of the transmitter molecules can reach the nearby postsynaptic receptors. But the acetylcholine concentration thereby falls immediately to a low level, and it falls not because of hydrolysis, but because the major portion of the transmitter packet is caught and very rapidly absorbed by the receptors. And then the subsequent dissociation is a much more gradual process, extending statistically over a few milliseconds, and during this period, free acetylcholine molecules emerge asynchronously in low concentration and are well taken care of by the esterases. But this, incidentally, is not the complete story. There's more to it. The hydrolysis of acetylcholine serves a twofold purpose. It not only stops postsynaptic receptor activation and leads to a rapid restoration of the membrane potential of the muscle fiber, but it also provides choline for a highly specific uptake system in the terminal, and so it aids the recycling and intraterminal resynthesis of the transmitter substance. Now you might say that the cholinesterase is a very special case at the neuromuscular junction. At other synapses we have different methods of inactivation or disposal of the transmitter molecules. And yet the problem is much the same. At all synapses there's bound to be some degree of competition between the inactivation sites and the postsynaptic receptors. And the Quantal release, the focal release in very high concentrated packets, seems to be specially designed to ensure access of the transmitter molecules to the receptors by oversaturating and so initially bypassing the inactivation sites. Well, now I come to the last aspect I want to discuss. When we first recorded miniature end plate potentials, we found that their amplitude showed an interesting and clear correlation with fiber size. The smaller the muscle fiber, the larger was the amplitude of the miniature potential. In fact, what helped us in the accidental discovery of the whole phenomenon was the peculiar circumstance in which Paul Fett and I happened to search for slow muscle fibers. This caused us to turn to a toe muscle, whose fiber diameters are on the average considerably smaller than in the sartorius, and this was a great help. 6 years later, Stephen Teslev and I made a systematic study of this inverse relation between fiber diameter and miniature size. And we confirmed that there is a very strong correlation, the amplitudes varying 5 to 10-fold between the largest and smallest fibers that we could handle. Our conclusion then was that the miniature potential resulted from the release of a relatively constant quantum of acetylcholine, whose reaction with the postsynaptic receptors opened about 1,000 ion channels, and so produced a fairly constant conductance increase, a more or less constant shunt across the postsynaptic membrane. The smaller the fiber, the higher its input impedance, and therefore the larger the depolarization produced by a given short-circuit pulse. Now, if one looked at it more closely, one found that the relation between miniature potential and input resistance departed from direct proportionality. There seemed to be a limitation on the size of the potential reached in the very smallest fibers, up to several millivolts, so that regardless of cell size, there always remained a large safety margin between these spontaneous local depolarizing blips and the firing threshold of the fiber. Well, this is a point which has been emphasized and brought out more clearly in a very interesting recent paper by Dr. Wilkinson and his colleagues. Now, the question of regulating the size of the quantal response of the miniature synaptic potential acquires a much greater, indeed vital importance at central synapses, because here it is not just a matter of keeping spontaneous blips well below thresholds. At central neurons, whose function depends on the integration and mutual interaction of multiple afferents, the effect of one quantal packet has to be kept small enough not to interfere with the integrated function of the converging inputs. At a neuromuscular junction, a vesicle releases several thousand transmitter molecules, which open 1,000 ion channels and produce an inward current of several nanoamps. And that is in a fiber of, say, 500,000 ohms input resistance. Now, a similar action impinging on a small granule cell in the central nervous system with an input resistance which may be several hundred or even 1,000 times greater, this would be quite intolerable. It would totally depolarize and probably destroy the cell. Recent investigations with the patch clamp technique have thrown some very interesting light how the necessary regulation is achieved. It appears from work done in several laboratories, including those of Bert Zachman, Stuart Kulkarni, Tomoyuki Takahashi, Francis Edwards and their colleagues, it appears that in the different neurons of the central nervous system, the number of ion channels which are opened postsynaptically by a quantal packet of transmitter is adjusted in accordance with the size and input resistance of the neuron. Thousands of channels are opened during a miniature synaptic potential in giant synapses of the squid or lamprey, or at neuromuscular junctions of large muscle fibers. On the other hand, only a few hundred channels respond to a transmitter quantum in sympathetic ganglion cells, and only 10 to 20 channels in the small granule cells in the cerebellum and hippocampus. It's still not quite clear whether the size of the quantal packet of transmitter molecules plays a part in this process of regulation, or what seems in fact more probable, that the main regulating factor is the number of postsynaptic receptor molecules facing individual release sites. It rather looks as though at some of these synapses the limitation of the quantal response is brought about chiefly by presenting a small patch of receptors to a fairly large and possibly saturating quantal pulse of transmitter molecules. Now, the whole thing is a fascinating subject which remains under vigorous study. It will be of great interest to explore it further and examine the developmental stages of the regulatory process and find out how this vitally necessary adaptation of response size in relation to cell size comes about. Well, I've finished. May I emphasize what I indicated at the beginning of this report? I have derived many years of first-class entertainment from my work, and I'm very happy to see how our younger friends are advancing the field beyond anything that seemed feasible not so long ago, and how they are quite obviously deriving much further pleasure in doing so. Yes.
You will know from the Congress newsletter that Professor Ito has been elected to be the next President of the Union, and it gives me great pleasure to ask Professor Ito to propose the vote of thanks to Sir Bernard Katz.
After listening such a lucid, illuminating, inspiring lecture by Sir Bernard Katz, any word would be just superfluous. Deep impression is lingering on. Sir Bernard's Quantal secretion story is an outstanding landmark of physiological sciences, and we are fortunate today to have been able to listen from Sir Bernard himself. On behalf of the physiological scientists gathering here in Glasgow from all over the world, I express here our sincere thanks and admiration for Sir Bernard's great achievement, which symbolizes the success of modern physiological sciences. Thank you very much, Sir Bernard.